The Pathfinder Volume 49, Writing a Successful CMC Section for a Biologics License Application.
In this audiocast, we discover the secrets to a successful biologics license application in this captivating guide. Delve into the heart of the application process and unlock the power of the chemistry, manufacturing, and controls section. Unleash your potential by understanding the intricate details of the manufacturing process, quality control, and analytical methods behind your biologic product. With this knowledge, you can easily navigate the treacherous waters of regulatory approval, avoiding delays and rejections. Trust the process and give yourself the essential tips and insights to craft a compelling CMC section that will captivate regulatory reviewers. Don't settle for mediocrity. Seize this opportunity to make your mark in biologics. The power to succeed lies within your grasp. Are you ready to unleash it? Check out this audiocast.
Developing a successful biologics license application requires significant effort and attention to detail. One of the most critical sections of the BLA is the Chemistry, Manufacturing, and Controls section. This section details the manufacturing process, quality control, and analytical methods used to produce the biological product. A well-written CMC section can help to ensure a smooth regulatory approval process and reduce the risk of delays or rejections. To write a successful CMC section, there are several essential things to consider before you start authoring. First, you must thoroughly understand the manufacturing process and quality control procedures used for the biologic product. This includes knowledge of the raw materials, manufacturing equipment and processes, and analytical methods for testing the final product. Second, you must clearly understand the regulatory requirements for the CMC section. The U.S. Food and Drug Administration has strict guidelines for the content and format of the CMC section. These guidelines must be followed closely to ensure the CMC section is comprehensive and meets all regulatory requirements. Third, you should have a solid understanding of the scientific principles and concepts underlying the production of the biological product. This includes knowledge of the cell bank and process of the product, as well as any changes that occur with the product during development. In drug development for biologics, the phrase "the process is the product" means that the manufacturing process used to produce a biological product is integral to the final product itself. Unlike small molecule drugs, which can be chemically synthesized and have well-defined structures, biologics are complex molecules often produced using living cells or organisms. The manufacturing process for biologics involves numerous steps and must be carefully controlled to ensure that the final product is consistent and meets the necessary quality standards. Any changes to the manufacturing process can potentially impact the safety, efficacy, and quality of the final product. As a result, the FDA places great importance on the process used to manufacture biologics, which is considered a critical part of the final product. This means that manufacturers must carefully control and document the manufacturing process for biologics, and any changes must be carefully evaluated and justified before they are implemented. Once you have a solid foundation of knowledge and understanding, you can begin authoring the CMC section. Here are some basic but essential tips to keep in mind. Use a clear and concise writing style. The CMC section should be written clearly and concisely, using language that is easily understood by regulatory reviewers. Organize the section logically. The CMC section should be organized in a logical and easy-to-follow manner. This can help reviewers to quickly and easily find the information they need. Provide detailed information on the manufacturing process. The manufacturing process is a critical component of the CMC section. You should provide detailed information on the manufacturing process and document things early, including descriptions of the equipment used, the manufacturing steps, and the quality control procedures. Describe the analytical methods used to test the product. The CMC section should include a detailed description of the analytical methods used to test the final product. This should include information on the accuracy, precision, and reproducibility of the methods, and changes over development should be bridged and documented. Discuss any potential impurities or contaminants. The CMC section should include a discussion of any potential impurities or contaminants that may be present in the product. This should include a description of the steps taken to minimize the risk of these impurities or contaminants. Include all necessary documentation. The CMC section should include all necessary documentation, including batch records, stability data, and analytical data. Once again, start early in development. While there are many similarities between the CMC section of a biologics license application and a new drug application, there are also several key differences. One of the most significant differences between the BLA and NDA CMC sections is the complexity of the manufacturing process. Biologics are typically more complex to manufacture than small molecule drugs, which means that the CMC section of a BLA is typically more detailed and complex than that of an NDA. The CMC section of a BLA must provide a thorough description of the manufacturing process, including information on the cell lines used, the purification process, and the analytical methods used to test the final product. Additionally, the CMC section of a BLA must include data on the stability of the product over time. Another key difference between the BLA and NDA CMC sections is the regulatory requirements. The U.S. Food and Drug Administration has specific guidelines for the content and format of the CMC section of both BLAS and NDAS. However, the guidelines for BLAS are generally more extensive and more stringent than those for NDAS. Biologics are typically used to treat more severe or life-threatening conditions, so the FDA requires more scrutiny for biologic products. Despite these differences, several critical similarities exist between the BLA and NDA CMC sections. Both must describe the manufacturing and quality control procedures used to produce the product. They must also provide information on the analytical methods used to test the product, including accuracy, precision, and reproducibility. Additionally, both the BLA and NDA CMC sections must provide data on the stability of the product over time. As you build a BLA, there are several fundamental CMC areas that you should focus on to ensure a successful submission. Manufacturing process. A detailed description of the manufacturing process is a critical component of the CMC section. This should include information on the cell lines, fermentation or culture, purification, and final formulation. You should provide a detailed description of each manufacturing process, including details on equipment, reagents, and process controls. Quality control. Quality control is another critical area of the CMC section. You should provide a detailed description of the quality control procedures used to ensure the final product's safety, purity, and potency. This should include information on the methods used to test for identity, potency, purity, and impurities, and the acceptance criteria for each test. Analytical methods. A detailed description of the analytical methods used to test the final product is another key area of the CMC section. This should include information on the method's accuracy, precision, and reproducibility. You should also include data on the validation of the methods used and any reference standards used in the testing. Stability. Stability testing is a critical component of the CMC section. You should include data on the stability of the product over time, including information on the storage conditions and the testing intervals used. Stability data should be presented clearly and concisely, and should include data on the degradation products and impurities observed during the stability testing. Comparability. Comparability testing is an important area of the CMC section, particularly for biologics that have changed to the manufacturing process or formulation. You should include data on the comparability of the product before and after any changes to the manufacturing process or formulation and provide a justification for any observed differences. Container closure system. The container closure system is another key area of the CMC section. You should provide a detailed description of the container closure system used, including information on the materials used and the testing performed to ensure the integrity of the container closure system. The manufacturing process is a critical component of the CMC section of a BLA, and a detailed description of the manufacturing process must be included in the application. The manufacturing process information should be presented clearly, concisely, and well organized to ensure that it is easy for the regulatory authorities to understand. The following are some of the critical pieces of information about the manufacturing process that should be included in a BLA. Description of the process. A detailed description of the manufacturing process should be provided, including information on the cell lines used, the fermentation or culture process, the purification process, and the final formulation. The description should include step-by-step details of each process, including information on equipment, reagents, and process controls. Process development. The BLA should also include information on process development and optimization. This may include details on the cell line selection, the fermentation or culture process optimization, and the purification process. Process controls are critical to ensuring the quality of the final product. The BLA should include a detailed description of the in-process controls used to monitor the manufacturing process and ensure that the product meets the required specifications. Manufacturing facilities. The BLA should also include information on the manufacturing facilities used to produce the product. This may include details on the equipment used, the facility design, and the environmental controls in place to ensure product safety and quality. Quality control. The BLA should describe the quality control procedures used to ensure the final product's safety, purity, and potency. This should include information on the methods used to test for identity, potency, purity, and impurities, and the acceptance criteria for each test. Batch records. Batch records provide a detailed record of each manufacturing batch and are a critical component of the CMC section of the BLA. The BLA should include a description of the batch record format and the information contained in each batch record. The analytical methods used to test the final product are a critical component of a Biologics License Application CMC section. A detailed description of the analytical methods used and the specifications required by the FDA should be included in the BLA. Here are some of the critical pieces of information about analytical methods that should be carefully documented early in development and then included in a BLA. Method validation. The BLA should include information on the validation of the analytical methods used to test the product. This should include details on the method's accuracy, precision, reproducibility, and reference standards used in the testing. Method development. The BLA should also include information on analytical methods development. This may include details on the selection of the method, optimization of the method, and evaluation of the method's performance. Specificity. Specificity is the ability of an analytical method to detect the product of interest in the presence of other substances. The BLA should include data on the specificity of the analytical method, including information on any potential interference from impurities or other substances. Accuracy. Accuracy is the closeness of the measured value to the true value. The BLA should include data on the accuracy of the analytical methods, including any biases or errors observed in the testing. Precision. Precision is the reproducibility of the measured value. The BLA should include data on the analytical method's precision, including information on the repeatability and intermediate precision of the methods. Limit of detection and quantification. The limit of detection is the lowest concentration of the analyte that can be reliably detected, while the limit of quantification is the lowest concentration that can be reliably quantified. The BLA should include information on the LOD and LOQ of the analytical methods used. Reference standards. The BLA should also include information on the reference standards used in the testing, including information on the source of the standards, their purity and identity, and their storage and handling. Here is a list of the baseline specifications that are typically required by the FDA. Identity and purity, potency or activity, quantity, biological activity, sterility, endotoxin, appearance, pH, osmolality, container closure integrity. The CMC section of a biologics license application is critical to the approval process of a biological product. Here are some of the most common CMC issues that can lead to non-approval of a BLA. Inadequate process validation. The FDA expects the biologic manufacturing process to be fully validated, with all critical steps controlled and monitored. Inadequate process validation is a common reason for non-approval and can result in concerns about product quality, consistency, and safety. Lack of adequate control strategies. The FDA expects manufacturers to have a robust control strategy to ensure product quality and consistency. A lack of adequate control strategies can lead to non-approval, as it can raise concerns about the consistency and safety of the product. Inadequate characterization. Inadequate product characterization can lead to non-approval. The FDA requires a comprehensive characterization of the product, including its structure, purity, and potency. Inadequate characterization can lead to concerns about the safety and efficacy of the product. Inadequate analytical methods. Analytical methods used to test the product are a critical component of the CMC section of the BLA. Inadequate analytical methods or a lack of validation data for the methods can raise concerns about the reliability and accuracy of the testing. Issues with impurities. Impurities in the final product can lead to concerns about the safety and efficacy of the product. Non-approval can occur if the level of impurities exceeds regulatory limits or if the impurities need to be adequately characterized. Issues with container closure systems. Container closure systems used to store and deliver the product are also a critical component of the CMC section of the BLA. Issues with container closure systems, such as inadequate or insufficient testing or lack of validation data, can lead to concerns about the stability and sterility of the product. Manufacturing facility issues. Non-approval can also result from issues related to the manufacturing facility. These may include inadequate facility design, lack of appropriate equipment and infrastructure, or failure to maintain proper environmental controls. The FDA has several CMC guidelines for developing and writing a biologics license application to approve a biological product. Some of these guidance include guidance for industry, content of the CMC section of an application or submission for a biological product. This guidance provides recommendations for the content and format of the CMC section of a BLA submission. It provides specific guidance for each CMC section, including drug substance, drug product, and analytical methods. Quality considerations in demonstrating biosimilarity of a therapeutic protein product to a reference product. This guidance provides recommendations on the CMC information that should be provided in a BLA submission for a biosimilar product. It guides the data that should be provided to demonstrate biosimilarity between the biosimilar product and the reference product. Comparability protocols for human drugs and biologics, chemistry, manufacturing, and controls information. This guidance provides recommendations for developing and submitting comparability protocols for human drugs and biologics. A comparability protocol is a written plan for assessing the impact of a proposed change on a biological product. Q5A Viral Safety Evaluation of Biotechnology Products Derived from Cell Lines of Human or Animal Origin. This guidance provides recommendations for evaluating the viral safety of biotechnology products derived from cell lines of human or animal origin. It includes recommendations for viral testing, clearance studies, and evaluation of potential viral contaminants. Q5E Comparability of biological products subject to changes in their manufacturing process. This guidance provides recommendations for the evaluation of the comparability of biological products before and after manufacturing changes are made. It includes information on the types of changes that may require a comparability assessment and the types of data that may be required to support the assessment. These are just a few of the FDA CMC guidance available for developing and writing a BLA. Other guidances are available on the FDA website and should be reviewed thoroughly to ensure a complete and successful BLA submission. The FDA's CMC guidance documents include topics such as manufacturing and controls information in an NDA, ANDA, or BLA, guidance for industry, chemistry, manufacturing and controls guidances, chemistry, manufacturing and controls changes to an approved application for certain biological products, chemistry, manufacturing and control information submitted in an IND, providing regulatory submissions in electronic format, certain human pharmaceutical product applications and related submissions using the ECTD specifications, comparability protocols for human drugs and biologics, analytical procedures and methods validation for drugs and biologics, comparability protocols for post-approval changes to the chemistry, manufacturing, and controls information in an NDA and/or BLA, considerations for the development of chimeric antigen receptor CAR T-cell products. These guidances are designed to help sponsors understand what is required when submitting a BLA. They guide how to develop CMC information that meets regulatory requirements. Additionally, they provide recommendations on ensuring the product is safe and effective. By following these guidances when developing a BLA submission package, sponsors can be confident that their product will meet all necessary regulatory requirements. This will help ensure that their product is approved quickly by the FDA. Speaker 3 in conclusion. The CMC section of a biologics license application plays a critical role in the regulatory submission process for biologics, providing the FDA with a comprehensive understanding of the manufacturing process, analytical methods, and product specifications for a proposed biological product. Manufacturers must focus on critical areas such as process development, analytical methods, specifications, and stability testing to ensure a successful BLA submission. They should also consult relevant FDA guidances for biologics to ensure that their CMC section meets the content requirements and expectations of the FDA. By following these principles and guidelines, manufacturers can optimize the chances of obtaining FDA approval for their biologic products, ultimately helping to improve patient health and well-being. The manufacturing process used to produce the final product is considered an essential component of the product itself. This is because the properties and quality of a biological product are primarily determined by the manufacturing process used to produce it. Therefore, in a sense, the process is indeed part of the product. For example, the manufacturing process can impact a biologic product's purity, potency, and safety. Any changes to the manufacturing process can potentially impact the product's quality, safety, and efficacy. As a result, the FDA places great importance on the manufacturing process for biologics, and it is a critical component of the regulatory submission process for biologic products. There is no single most crucial CMC information when writing a BLA. Instead, the CMC section of a BLA should be viewed as a comprehensive and interconnected set of information that must be presented clearly and thoroughly. However, some critical components of a thriving CMC section include a detailed manufacturing process and control description, analytical methods, specifications, and stability testing. In addition, it is essential to consult relevant FDA guidances for biologics to ensure that the CMC section meets the content requirements and expectations of the FDA. By focusing on these critical areas and following FDA guidance, manufacturers can optimize the chances of obtaining FDA approval for their biologic products. Finally, the CMC section of a BLA is a complex and critical aspect of the approval process for a biological product. To avoid common CMC issues that can lead to non-approval, it is essential to ensure that the manufacturing process is fully validated, that adequate control strategies are in place, that the product is well characterized, that appropriate analytical methods are used, and that issues with impurities, container closure systems, and the manufacturing facility are addressed.
Thank you for listening to this audiocast. Are you an emerging biotech or established pharmaceutical company struggling to keep up with the ever-changing landscape of drug regulations? Look no further than Incrisi. We are the leading consulting group helping organizations comply with drug regulations through a comprehensive drug development model incorporating augmented intelligence and data analytics. Our industry experts can guide you through the complex world of compliance, making it more manageable than ever. With our Reimagining Regulatory Pathways services, we can help you design and implement best practices to streamline your operation and ensure you stay in line with current legislation. Our range of services covers regulatory drug development's technical and strategic aspects. Let us help you develop an overall strategy that accounts for all potential risks while still being able to meet your objectives. Visit our website at www.enkrisi.com to learn more and get started on the path to regulatory compliance success.